What I'm going to be talking about today is soy glaceolins and how they regulate transcript abundance in the female mouse brain. Glaceolins are produced by soy plants in response to stress. They have anti-estrogenic activity in breast and ovarian cancer cell lines in vitro and vivo. Several recent studies have reported that mixtures of the Insomnia's glaceolins 1, 2, and 3 affect mammalian physiology in vivo and in vitro. And what emerged from that theory is a complicated and incomplete picture of how GLI affects mammalian physiology. The findings also indicated that GLI opposes the activity in estrogen-responsive tissue, such as the uterus. An example of this would be GLI inhibited E2 stimulated growth of MCF7 breast cancer and BG1 ovarian cancer cells. They implanted as xenograft tumors in female mice that have no uterus in vivo. However, other studies have concluded that GLI can also exhibit weak estrogenic activity, which appears to be enabled GLI to promote the proliferations of MCF7 breast tumor cells in vitro. Glaceolins exhibit mechanisms of actions not involving estrogen receptors, also known as ER signaling. In this study, the scientists implanted slow-release pellets, also known as the placebo, into female mice that have had their ovaries removed, which was followed by 11 days of exposure to GLI or vehicle IP injections. The scientists then performed a microarray on total RNA extracted from whole brain hemispheres and then identified differential expressed genes by a 2x2 factorial ANOVA with a false discovery rate of 0.20. Here's a chart, and what this chart means is the heat map shows the estrogen groups often respond opposite to the glaceolin-only group, and when both are used together, the pattern on the chart looks like the estrogen group and the GLI group. That means that the estrogen is the stronger signal. Each column represents one female mouse, and each row represents a gene. Gene expression is measured by the amount of mRNA for that gene in the brain. The average mRNA expressed for that gene across all groups is in black. Red indicates increased expression and also meaning that there are more mRNA copies are produced for that gene. Green means decreased expression and also means fewer than average amount of mRNA copies. In total, the scientists identified 33 DEGs with a significant estrogen effect, 5 DEGs with a significant GLI main effect, 74 DEGs with a significant GLI and estrogen main effects, and then 162 DEGs with a significant interaction term. When going over all DEGs, it revealed the transcript abundances were similar between the estrogen and the GLI and only estrogen treatments. However, after GLI-only treatments, the gene expression was distinct from both of these treatments and was characterized by higher transcript abundance. The results suggest that whether GLI acts in the brain through ER-dependent or ER-dependent mechanisms depends on the target gene.